So our second speaker is Dr. Klaus Gramann. Dr. Gramann is the chair of psychology and neuroergonomics at the Technical University of Berlin. Since 2017, he has also been a professor at the School of Software, University of Technology in Sydney, and his research covers the neural foundations of cognitive processes with a special focus on brain dynamics, on the brain dynamics of embodied cognitive processes. And today he will be talking about imaging natural cognition in the real world.
Thank you very much. Thank you very much for the invitation. I'm super excited to be here. Unfortunately, I'm super tired also because I got laid in, so my language might be sluggish. More dangerously, my thoughts might be sluggish too. However, I'm going to talk about a new modality. Now, we've been talking about fNIRS for most of the time, and I think I thought I'd provide you the perspective from EEG as a mobile brain imaging modality, and how we use that. Some of the questions that have been discussed already, and why I think it's really important that we use mobile brain imaging approaches, because things will differ. And this is very obvious, and everybody in this room knows that. That's why we're here. So I'm praying to the choir. But the moment that an animal changes its behavioral state, that is when the brain dynamic state also changes. And this is by definition the case because movement leads to sensory feedback that will be processed by the brain, and thus the brain dynamics will differ, period. So the ground truth that we're talking about when talking about brain activity in the scanner or seated in front of a screen fixation cross, that ground truth might be a ground truth from what we know in established brain imaging studies, but not necessarily what the brain has evolved to do. That would be optimizing the outcome of our behavior. And we are Dickinson basically state that the architecture, the neuroarchitecture obviously remains the same more or less. The connectivity patterns changes immediately when animals change their behavioral state. Nonetheless, what we do traditionally is put people in a scanner, lying supine, watching something, or sitting in dark rooms, fixating a fixation cross on the screen, and then we take the millions of bits of information that we get per trial from these imaging approaches, and we compare that with 1 bit of activity of behavior, that is the button press at the end of the trial. And there is a dimensional mismatch of what the brain has evolved to do. So this arguably is not the right approach, for me at least, to understand the human brain. Then the question obviously is, we've been doing these traditional brain imaging approaches for decades now because there are some restrictions, like it's difficult to push a 3-ton scanner around following the subjects, or looking at analysis approaches that are compromised by muscle activity and eye movements when we consider those kind of biological activity patterns artifactual. Computer power allows us now to analyze the signals differently, and then the question is, how can we overcome the restrictions? And that's pretty straightforward. We all know that we need some kind of modality to image brain activity. At the moment, that would be EEG, mobile EEG, functional near-infrared spectroscopy, potentially optically pumped MEG in the future. I've seen a PET scanner, a mobile version, obviously not the best solution, but there is a strong development. If you want to understand how the brain controls and is influenced by behavior, however, we have to synchronize that with active behavior. So we have to couple that with motion capture or other kinds of behavioral measures. And if we are interested in the entire context, if we want to understand what else impacts brain activity and how the brain impacts behavior, then we need, for example, eye tracking in head-mounted virtual reality. We use virtual reality because we have control over the protocols. But the same idea is true also for real-world neuroimaging. We do the same things in the real world, not necessarily only in the lab. This is a lab that you see here, the Berlin Mobile Brain Body Imaging Lab in the background. We have 150 square meters dedicated to virtual reality and real-world navigation or any kind of other experiment, where we synchronize EEG with eye tracking, motion capture, ECG, EDA, and other modalities. And what I would like to see, or what I hope to see in the near future, would be a coupled system of EEG and fNIRS, obviously. But that is not trivial, as you all know. So synchronizing data streams wirelessly, because we don't want to impact the movement range of participants, is difficult. They come in with different sample rates, so tons of technical issues we have to overcome. Bluetooth disasters we experience every single day in the lab. This is non-trivial, but there are solutions, and I'm pretty sure they will improve over the next years. LSL is one of the solutions, lab streaming layer. I don't know who else is, what other software architectures are used. That would be some of the things I would like to learn here. And then obviously, and that for me, like for David, is like the bigger question, how do we analyze that data? And I'm coming back to that in the discussion, because what we do currently in this kind of mobile brain imaging is a very brain-centric approach. We use events from behavior, events that we provide stimulation to then extract brain responses, but obviously this also goes the other way. And we're talking about a predictive brain that might impact behavior differently than we might assume. This is a pipeline that we've been working on over the last 10 years. And I think one of the other aspects that I would like to propagate here is exactly that. Sorry for that. That this kind of new era in mobile brain imaging should start with standardization and open data. We should share protocols, we should share approaches, and be able to replicate all the results that we get, because it's difficult enough to publish the results, because we have more critical views on the data than the community has people to work on. So besides synchronizing mobile different modalities and analyzing all these multimodal data, obviously we need data-driven approaches. Alex has shown that. For EEG, and that's a special case, it's not so much the case for FNIRS, but some of the problems hold true too, is that if you look at EEG signal, this is a volume conducted signal, it's an ill-post map problem, we cannot resolve the sources, and what we get, it's like a point spread function. If you, if you have two sources of alpha activity 9 and 11 hertz here in the left motor cortex, right parietal cortex, this is slowed down, this is a realistic head model. So, and then you simulate this activity in these cortical regions, and then you back project that through that realistic head model to the surface where our sensors are. What you see is, first of all, you can't separate two different sources being active in parallel. We have tons of sources being active in parallel, and you will get activity from parietal cortex and motor cortex also in frontal areas. So this is one of the big issues that we have with EEG, and that might be more problematic than what you have with FNIRS. But with that kind of approach and other data-driven analysis approaches, we are able to dissociate brain from non-brain activity. This is one of the first mobile experiments that we did in 2007, together with Dan Ferrison back in Michigan. And here we have, again, like multimodalities, EMG, motion capture, force measures, EEG, obviously. And if you consider volume conduction of only brain activity, and then you think about these kind of setups, then this was like my 3-year postdoctoral nightmare. I still dream of the data. This is what the data can look like then, obviously, when people are running on a treadmill. However, if you get the movement information, gait cycle, heel strikes, head movement, and all these things, then you can reconstruct. In this case, we used ICA. You can reconstruct brain activity and dissociate that from non-brain activity. And I'm specifically saying that non-brain activity is not necessarily artifactual. In EEG research, neck muscle activity, supracranial muscles, eye movement is considered artifactual, but it's not. It's part of the cognitive system. It helps us to solve a task. If I'm running, and I have to stare at something that approaches me, I have to compensate the head movement in 3D space. That is done with eye movement and neck musculature. So that's part of the cognitive system to make sure that that fly doesn't hit my face. So why would we then get rid of that rather than analyzing it together with EEG activity, like other modalities? And this is what we do. And then you can even proceed and try to get more realistic head models. not model only the brain, but use EMG, use muscles, use eye movements, and get individual head models. Obviously, that costs money, costs time, but that would be a step forward to a more accurate description of what is going on in the human brain during activity. And I would like to show you one example that, from my perspective, shows very strongly why it is important to have active behavior. This is a simple spatial cognition tag. It's a heading computation. So again, decades of spatial cognition research in the scanner has shown like marvelous results. And in all of these studies, participants navigated based only on visual information. If I close my eyes and I turn around, I still know that you're somewhere there, based only on proprioception and vestibular information that is used automatically to update spatial position and orientation. You cannot undo that. That is a neurosignal. The second order vestibular system is already multimodal. So if you then basically create a simple paradigm, and this touches on this kind of experimental protocol discussions that we had, and how do we get ground truth? Then basically, in this case, we used a spot rotation test. It's not really mobile yet, right? People are just standing on the spot. They see an orienting beacon that's replaced by that sphere, rotating at different velocities around them at a given distance, and stops at an unpredictable eccentricity on each trial. And then it changes its color, you turn back, and then you indicate your original heading. And with that, we get like over and underestimation, we get the velocity profile, outward, backward rotation, all of the behavior that we get. And this is the crucial part here. Now we fall back to our ground truth. This would be the standard experimental protocol in traditional brain imaging, where participants control the movement with the joystick rather than with the full body. And then we do within, so it's a repeated measure, and that's important. It costs a lot of time, 3 hours, that experiment. People were really tired. And then we compare basically both of these experimental conditions to see what are the changes in brain dynamics in active behavior as compared to traditional setups. The behavior itself, this is the reproduction error, the heading reproduction error, or as a function of increasing eccentricity. And in blue, you see the joystick condition. So you see a monotonic, more or less monotonic increase with increasing eccentricity of that heading reproduction. If you have participants move naturally, that basically is a drastic difference. First of all, they are way better. The heading estimation is more accurate. Second of all, you see that there is a main effect of eccentricity, but there's also an interaction. And that is quite interesting because up to 90, 100 degree, there's not much of an increase. But when you start rotating beyond your cardinal body axis, something seems to switch in the cognitive processes going on to update your orientation in space. So more interestingly, obviously, is what does the brain look like during these kind of tasks? And to answer that, we look at the muscle first. So this is an ICA decomposition. Excuse me. Single ICs, small spheres here, clustered using a repetitive k-means clustering approach with the region of interest. And the big sphere here is the centroid of that cluster, multidimensional k-means clustering. This is in a position that likely reflects the sternocleidomastoid muscle that was active or is active whenever you rotate your head and down. So what we did here is we looked at the activity pattern of these ICs across all participants, and we did that by computing the velocity continuously during the outward rotation. And then we binned that 10 percentiles from the lowest to the fastest velocity. On a single trial, we computed band filtered Hilbert transform, and then we sorted that according to the velocity. This is a standard protocol that was already used in rat identify orientation behaviors by Taube in 2001. And then basically what you see here, if you sort the different power values according to the velocity within each frequency band, you might see this kind of a striping behavior that would indicate that there is a change in power in a specific frequency band in that region, maybe muscle, brain, or eye that is dependent or manipulated by the velocity. That was the first step. You do that for all the different frequencies. And then we computed a Mahalanobis distance between all the dimensions that we had here. So it's like multi-dimensional distance measure, and we created a dissimilarity matrix. And with that, basically, you now have all your lowest to highest velocities, the different power values for the spot rotation only in the upper left part, the direct comparison of the stationary and the rotation, the physical rotation, or the joystick condition only. So this is a dissimilarity matrix, and then you do permutation testing on that. That's a 1,000 repetition permutation with 0.05 significance level. And this is the entire range. And this is the real data for the right neck muscle. And what you see then here is that in the physical rotation condition, there is a clear velocity-dependent modulation in different frequency bands, dependent on the velocity, which is more or less suppressed in the joystick rotation. And this is clearly visible in the mobile rotation. There's massive differences between mobile and stationary, and there is nothing going on in the stationary condition. And if you run the statistics, that is more or less confirmed, because in the stationary condition, you don't rotate your head, you just rotate using a joystick. There is no head movement. And this for us was kind of a sanity check. And there's tons of really interesting results in this paper, but I'm going to show you, for me, the most interesting one, and that is the visual cortex in this very same task. And what the visual cortex basically shows here, this is a region, a cluster located in or near BA17 at the border to BA18. In or near, it's EEG. I know we are just estimating that as an approximation. But what you see is there's not much going on in the physical rotation condition dependent on the velocity, but there are changes dependent on the velocity in the joystick rotation condition. And you see the same pattern that you saw for the muscle, but inverted. Tons of difference between the two conditions, with differences in the joystick, but no differences in the active rotation condition. You know what kind of sensory information you will receive when you rotate actively. This is predictive processing. Your visual cortex does not have to process anything that's going on other than the sphere that's moving. And coming back to that, the question is that ground truth that we compare our data to? Or did we just establish a stationary, to a certain degree, compensating mechanism that tries to deal with sensory conflict? Because your visual system tells you you're rotating, but vestibular and proprioceptive information clearly tells you, no, you're not. And by the way, you're lying in a scanner on the back. So that is one of the questions that we should ask ourselves, and that we have to, I think, communicate more clearly. So I tried to gather a few questions for this session that I have. But summarizing here is that if we start moving and actively behaving, that behavior will provide sensory feedback that is processed by the brain that will, by definition, change the brain dynamics that we record. If we then go into the real world, we lose control over tons of factors that now impact our results. And this is something that we already did. We can now start using all these new kind of information that you get from movement, but also asking yourself, what kind of events can we use if we have no control over stimulation? And one of the aspects that we can do in this case for EEG is using blinks. Every time you blink, you close your eyes. Every time you open your eyes, you have transient visual information. And since the 60s, it has been shown that the number of blinks is significantly higher than it would be necessary for moisturing your eyes. It correlates with mental states. And it clearly reflects cognitive activity. So why wouldn't we use these kind of informations to now start diverging into different directions to create new events that we can use from these kind of new environmental, new experimental environments that we have? So this all will change dynamics as we know them. The questions that I have, or the summary is, behavioral state changes the brain dynamic state, I'm pretty sure that we will replicate a lot of the early evoked activities, because these are functional in the sense of incoming information that will be processed more or less in the standardized way by the brain. But when we start looking into time ranges beyond 200, 300 milliseconds, and specifically the frequency domain, I think things will change drastically. And with that, I think mobile brain body imaging, irrespective of the modality that we use here, will be the new method to provide us with complementary insights to better understand human brain activity. We've been there, we have tons of questions. I have tons of questions, and I was super sad to hear that David was not able to answer how we analyze all the multimodal data. That was my hope to come here. But I think that is the super opportunity here to discuss that. So the first question is, what kind of protocols and analysis strategies are required for an objective, reliable, and valid research? And again, coming back, I think we have to provide standardized data formats, bits, including motion, including eye tracking, including all the modalities that we need. These are the formats that we have to develop to exchange data and to allow replication across labs. We need multi-lab studies, confirming basically from independent labs what we're looking for. How can we systematically rule out, this is, I think, one of the most important points, movement-related activity and/or artifacts, if movement is the very core of the research. And we have no ground truth, because movement and associated activity has never been investigated. So how do we get a ground truth? How can we make better use of high-dimensional and expensive data? Coming back to Joy's data, we have data that one PhD student can work on for 6 to 7 years. And it's all the data I showed you, the spot rotation, it's one paper that we published. And the data is just lying around, nothing happening anymore. So how can we improve that? How can we be more effective with these really expensive data sets? And are we able, or do we have to replicate established features? The question, do we need an MRI ground truth? Do I need a stationary comparison to say, this is what changes? From a publishing perspective, obviously we need that, because otherwise a lot of reviewers will basically say, no, I don't believe you. And the second part, last part, how do we systematically analyze multimodal data? And this is the question, if you look at cognitive processes, movement, EEG, this is like millisecond time range we're looking at. If we look at FNIRS, ECG, we're talking about seconds time range. If we look at EDA and other aspects, we're talking about minutes. How do we converge these data? How do we use the data to get more insights from multimodal than looking only at single modalities? And what I said, this brain-in or the CNS-centric view on analyzing data is the one that's currently the most dominant approach, where we use blings or like events that trigger cognition and then look at what happens in the brain. But we can also ask, and if you go to the interoceptive community, they're interested in what happens to your perceptual processes with a systole of your heart. It changes. You're more sensitive in sensory information processing during the diastole of your heart cycle. So we can also look the other way around, going more into potential physiological and movement-related impact on the brain dynamics and the other way. So analysis approaches, I have no idea. I would love to discuss with all the people that had way better experience and ideas about that. But cross-correlation, multifractal analysis, nonlinear dynamics, what kind of machine learning approaches? How can we address that with different analysis approaches? These are the questions that I would have. Thanks to a great team, like all of this research and everybody before has shown that. Tons of people that are involved and that do great work. And this is like the last question that I have. We have like this mobile brain body imaging starting in 2012, 13. And the communities are so far apart. So I see the necessity that we have to move closer together, that we have to combine these measures and combine forces to change politics in research funding, for example, but also why not thinking about our own journal? After the death of Neuroimage, I think it's a good time to think about things like that too. Okay, with that, thank you very much.
Virtual attendees, please type your questions, and in person, please come to the mic.
Thank you very much for a very interesting talk. And I'm wondering, of course, the multimodal measurement is really appealing, but what is the technical compromise and how many modalities do you recommend?
Well, the recommendation would be dependent on the research question you would like to answer.
I see.
My dream obviously would be everything that you can get on a person without compromising natural behavior, which is not the case anymore. If you look at 156 cables hanging down in the backpack with an amplifier and all the other stuff, that's not natural. But if you look at the development of the technical devices, wearables, we can get ECG activity constantly from millions of people that share their data using smartwatches. The same for GSR. And the development of mobile EEG and mobile fNIRS, I think, is very promising that might be small enough and lightweight enough that people will start using it at the moment that it brings an advantage for them.
I see. Thank you very much.
I thank you. I love the active incorporation of movement into the lab and that research. So this is a bit more of looking forward, but do you have any predictions or hypotheses that you would like to share or that you think you have in regards to what you'll see with more higher intensity movements and exercises rather than the slow rotational that you're working with at this point?
Yes, so that's a very good question. We just, I think, should be coming out in the next 2 weeks. It's a bioRxiv paper already where we looked at different movement intensity and the data pre-processing impact on ICA decomposition quality, looking at signal-to-noise ratio, the number of IC labeled brain and non-brain components, clearly shows that there seems to be a sweet spot that was unpredictable by us. So it doesn't even have to be like such a high dimension of channels. 64 channels seems to be a very good compromise already. And the biggest problems you will get in data quality is jerk. So a third derivative of the data. So the faster you accelerate, the more problematic it becomes with the artifacts. And that is a combination of tons cable sway, micro movement of electrodes, like Alex already talked about. So non-stationarity that you introduce to the data that ICA can't deal anymore. So yeah, so fast movements, rapid changes in movement direction are the most difficult things.
Yeah. Thank you.
Hi, thank you for a great talk. So I was wondering if kind of like the predictive processing discrepancies that you showed due to artificial type of tasks, if you could maybe speculate on whether you think that this would load on capacity limited resources of cognition, say working memory, attention. So my concern is that this could be potentially introducing a problem of load, of difficulty into cognition while we're trying to assess cognition, you see? So maybe I'm trying to measure working memory, but the artificial nature of the task and the prediction error that it provokes might create some distance between the construct I'm trying to measure and what I'm actually measuring.
Right, if I understand your question correctly, but then it would be more of a problem for a stationary protocol because you create a sensory mismatch that would be an overload next to your memory task.
Right. So I guess any artificial setup that would create a discrepancy, do you think that this would fit into the capacity resource, capacity limited problem?
Well, theoretically, yes. The question is, how high is the load in general, and do you reach capacity limits at that point with that kind of mismatch that you create in the protocol? I'm not so sure, and I'm also I think again, it's like for me it's converging evidence. So I'm not saying that everything that we have researched over the last decades is wrong. I think there's really important and truth in it, in the data, but we have to complement that with something that happens in a different way when we actually behave. And in that sense, yeah, there might be a difference. So it would be a nice protocol to directly compare the same working memory task during movement as compared to a stationary protocol. Maybe there is a difference.
Thank you.
Yeah, my question is not well formed, so help me out here. So your work is really beautiful and well matched with EEG. Have you thought about how, say, FNIRS could help expand the types of questions you're thinking about? And okay, I'm being selfish because I have a different technology, and I'm just trying to figure out experimental paradigms that we could use with the technology I work on.
I have not.
Yeah.
One of the reasons why I'm here, and one of the reasons why I hope that
Yeah.
Alex and I can do more in this direction. But I could imagine, for example, that all this integrated activity, if I created like a more difficult heading computation task, that we would get additional information about like the integrated load that starts to evolve over and outward rotation. Do we see differences if we looked at time watt volt changes dependent on the eccentricity? Would we see like a cross between 90 to higher eccentricities that would be visible in kind of FNIRS? But I'm too naive to the difficulties of designing protocols for FNIRS because I'm not an expert in the signal.
All right, we'll keep discussing.
Yeah.
Thank you very much.
Thank you.